All right, guys, so in this one, I'm going to be going over numbers 5 and 6 on this video. And this one is pretty intense on the diagram, but it's actually not too terrible once you kind of figure out what they're asking. So you might have seen this one in class. I think I gave it a long time ago, but it's a good picture to go over, as well as the questions are different. So this one says some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathway. Cells normally produce a steady supply of inactive caspase-1 protein, so that's right here, in response to intracellular pathogens. So intracellular means something's entering the cell. The inactive caspase is cleaved and forms an active caspase. So basically you have this protein, there's this molecule that's attached to it, and then it's removed once it's infected, and then you form an active caspase. Active caspase-1 can cleave into two other proteins. When caspase-1 cleaves, an active interleukin, step 2, the active portion of the interleukin is released from the cell. So basically, you don't need to know anything about interleukin, just follow the picture, and all you need to know is that this protein, this active caspase, can actually go ahead and activate this inactive interleukin and become an active interleukin. And then it just tells you that the active interleukin is released from the cell. Interleukin is a signaling molecule that can activate the immune response. So basically this just means that this interleukin, once it's activated, it leaves, and that's going to activate an immune response. Now let's see what the next portion is about. When caspase-1 cleaves, gastrodon, which is going to be right here, the N-terminal portions of several proteins associated with the cell membrane to form large non-specific pores. So this caspase go ahead and activates this gastromin. It's the N-terminal, so basically it just means it's the N-terminus part of the protein. And you don't even have to get that specific, just know that this is going to activate these pores. So it forms large non-specific pores as well. All right, so that's really what's going on. So bacteria infection activates the caspase, and it can activate those two proteins. All right, so let's see what the questions are about. So I just want to revisit this in terms of like cell signaling. Remind you guys, cell signal binds the receptor protein, it activates these proteins. And the big thing that I think a lot of people misinterpret is that all of these kinase proteins, all of these proteins in the transduction phase, they're already made in the cell. And so basically what I'm trying to emphasize here is that when you think of like a cell signaling pathway, the reason why they're so almost instantaneous is because all those relay proteins in the transduction phase, even this inactive, if we're looking at like a gene expression one, all these proteins are there ready to go. They just need to receive the correct signal, and then those proteins get activated and inactivated. So the reason why that's important for this one is just to kind of understand that all of these proteins, even though they're inactive, they're already made. They're just waiting for the right response, or I should say the right signal. In this case, what gets this thing going is the bacterial infection. So it is, for the most part, a cell signaling type of problem. So part A, describe the effect of inhibiting step 3 on the formation of the pores and the release of interleukin from the cell. Okay, so this one sounds more daunting than it actually is. So basically you're looking through this and it's saying that what if you stopped this pathway? What if this step was inhibited? What's going to happen? So the first part, let's answer that one. No pores would be formed. All right. So, and again, if you just stop that, if that step is inhibited in some way, shape, or form, you don't have to understand why. You just need to know that if that step was inhibited, you're not making the pores forming. So just reading through the animation, that's what I wanted you to get out of this. Now, what about the release of interleukin? Well, interleukin is over here. Step 2 is completely independent of step 3. So hopefully you're able to understand that interleukin is still released. Now, this one doesn't have a justify or response, but if it is, you just would state what I just mentioned, that step 2 is completely independent of step 3. So understand that. That's kind of pretty easy, simple 2 points. Let's move on to part B. For part B, it says, explain how cleaving inactive caspase 1 can result in the activation of caspase 1. Okay, so this one is kind of interesting, and you might be kind of freaking out about how to answer this. You wouldn't know. It's actually a lot more simple than you would think. So remember that the key thing on this one would be allosteric inhibitors / activators, right? So if I had, let's say, an enzyme here, and this right here is my active site. And remember that things can bind not at the active site, but allosterically. So what if I have this molecule bound to it? That would be essentially an allosteric inhibitor, right? Now, if all of a sudden, if I removed that allosteric molecule, and hopefully you're remembering this back now, you're kind of seeing where this question is going to be headed. If I remove that allosteric molecule, how is that going to look on my active site shape when it gets removed? Well, what if I do remove it? Well, maybe it can't actually change the active site shape when the allosteric inhibitor is removed. So if I say the inhibitor is removed, the active site shape changes. So basically this makes this active by removing that allosteric molecule. So how I would respond to this, explain how cleaving an active caspase can result in the activation of caspase 1. I would state in my answer that removing the inhibitor makes the caspase active. And I'll even put in parentheses due to changing shape of the active site. So that's how I would answer this one. It really was kind of dealing with enzymes and that allosteric inhibitor. So remember, inhibitors aren't necessarily innately a bad thing. Just understand that if you remove that allosteric molecule, you can change the active site shape, making that enzyme active. So that's part B. So let's check out C. Predict the most likely effect of a gas turbine pore formation on water balance cell in a hypotonic environment. Okay, so remember, hypotonic environment means that the cell is going to get fat like a hippo. Really cheesy but simple way to remember it. So it's a hypotonic state. So remember this hypotonic environment. So I would essentially state that if this is open and it's in a hypotonic state, water is going to want to rush in. So I will state for this one that water moves in. Okay, provide reasoning to justify your prediction of part C. So just again, remember that common theme about hypotonic versus hypertonic and water potential, all kinds of things that we can get into here. But just the simple thing is that water always moves to where there is more solutes. So that just basically means that there's going to be more solutes inside the cell. And we can even dive into this a little further, that this is going from one solute to when it's active, right? You cleave it off. Now all of a sudden you have two solutes. Same thing here. This was one solute. Now you have two solutes. You're actually creating an increase in solutes, causing water to want to rush in. So I could even divvy off of this and say that the cleaving proteins increases the solute in the cell, causing water to move in. So just kind of remember that water always goes to where there's more solutes, and you actually create more solutes in this state because you're cleaving off those proteins. So those were the 4 points for that one. I just wanted to kind of mention that and revisit cell signaling. Now let's do number 6 really quickly. Or yeah, number 6. I know it says 4 here. This was on the actual AP test, but 6 on the assignment that you're doing today. So this one is pretty simple. For some reason, RNA processing has been an FRQ topic every year. This year might be a little bit different, but let's kind of dive into this one and see what we got. The figure represents the process of the expression of gene X in a eukaryotic cell. So this whole thing they're saying is gene X. It tells you that the primary transcript in the figure is 15 kilobases long. So basically this thing right here is 15 kilobases in length, okay? So really this just means that it's 15,000 nucleotides in length, 15,000 A's, T's, C's, and G's on the gene, 15,000 AU C's and G's on the RNA transcript, okay? The mature mRNA, though, is only 7 kilobases in length, okay? So basically wants you to describe what's the modification that most likely results in the 8 kilobase difference in the length of mature mRNA. Identify in your response the location in the cell where the change occurs. So nothing's labeled here, and I just wanted to revisit this, and you guys usually knock this out of the park. Remember that exons are essentially the parts that exit the cell. So remember, exons are the coding part. Introns, remember, introns stay in, so introns stay in. They're basically non-coding. And describe the modification that has most likely resulted in the 8 kilobase difference. This is all called mRNA processing. So it's called splicing. So basically, to earn the point on this one, you just wanted to mention that this is mRNA processing. That's what you're seeing here. So for A, to earn that point, you had to state that mRNA is processed. Okay, and remember that those are the SNRMPs, the SNRPs, and the spliceosomes. Do that, right? So you SNRP it, you splice it. So the mRNA is processed via the SNRPs and the spliceosomes. Those are the two enzymes that do that, okay? And then you create your coding sequence. Now, the second part for A, identify in your response the location of where this change occurs. Well, it's eukaryotic, then it's really just identifying where is the DNA. All the mRNA processing takes place right here. It's measuring, if you look at this picture, it's all taking place in the nucleus. So the second part for A is going to be located in the nucleus. So all the RNA processing is going to take place, all right? Now for B, B says predict the length of the mature gene X mRNA if the full-length gene is introduced and expressed in a prokaryotic cell. Justify your prediction. Okay, so basically this means that we take this gene, this whole gene sequence here, we're putting in a couple of things here. Predict the length of the mature MX gene if the full-length gene, so the full length right here, is introduced into a prokaryotic cell. Justify your prediction. Well, this is something that is important. If you didn't catch this when we went over this class, on the right is a eukaryotic cell. They do that RNA processing. Prokaryotic cells do not do that because they do transcription and translation at the same time. So they make their mRNA, then they translate it at the same time as well. So there's no RNA processing. So I would state that if we introduce this whole length that is going to be 15 kilobases in length, in a prokaryotic cell, it's going to be 15 kilobases in length. Justify your prediction. I would say that progs cannot do mRNA processing. All right. So those are two pretty good examples of FRQs, two different topics, and I hope that helps. We'll see you guys in the next one.